is the denouement of our series of lectures on VDJ recombination, because that whole process of VDJ recombination culminates in the formation of an antibody molecule. And I've drawn for you up here an antibody molecule. And the antibody molecule consists of two identical heavy chains. And the two identical heavy chains are heavy chains because they're bigger. And I'll show you the heavy chains, little arrows. There's one here, and there's one there. And two identical light chains. And there are two identical light chains, one over here and one over here. Okay? And each of those chains, light chain and heavy chain, have in them a variable region, and the variable region has been drawn in black. So the variable regions are in black, right? Variable regions. And the colors, let's draw a red and blue arrow. Red and blue arrow correspond to constant regions. The red to the constant region of the heavy chain, and the blue to the constant region of the light chain. Okay, so these antibodies can have incredible variation. In fact, the variation is absolutely mind-boggling. And because these antibodies have to recognize all kinds of foreign antigens. And I'm going to show you exactly how we get to such huge numbers using VDJ recombination. It turns out that we mentioned that there was a heavy chain gene. And I'm going to draw for you the heavy chain gene. Okay, so here's a single heavy chain gene. And it's the germline heavy chain gene. All right, there's the germline heavy chain gene all the way across there. It's a long germline heavy chain gene. And let me just say germline heavy chain gene. Heavy chain gene. So the antibody is a protein, but now we've got a gene, germline heavy chain gene. And the germline heavy chain gene consists of a number of variable regions. I'm going to draw a few of them. Dot, dot, dot. Okay. And in the human, it turns out, every animal is somewhat different, but in the human, there are roughly 50 variable regions. Okay? In addition to that, way downstream, let's put a stagger mark down there, there are a number of D regions. And I'm going to draw a few of them. Dot, dot, dot. And so there are a number of D regions, and in fact, there are about 25 D regions in the human. D1, D2, and D25. Okay? So, downstream of that, there are a number of J regions. And in the human, there are 6 J regions. Dot, dot, dot. 6 J regions. Okay. J1, J2, and J6. Okay. So all of these are a little bit different. So I'm going to make them a little bit different. All right. So there's the heavy chain gene right up here. And as we described before, VDJ recombination picks one of the V regions, one of the D regions, and one of the J regions, and brings them together. All right? So there's a tremendous amount of diversity that's produced from this recombination event. Now, downstream from the J region, now I've run out of space, so I'm just going to curl it around. I'm going to draw a block corresponding to the constant region of the heavy chain. And of course, because the antibody has it in red, I'll also draw it in red here. So this is the constant region for the heavy chain. All right. And notice that I've drawn the V, the D, and the J all in black because they're going to end up encoding the variable region of the heavy chain, which is up here. So how many different variable regions can we get out of this single heavy chain gene? Well, for any one of the J regions, you can have 25 different D regions. But there are 6 J regions, each of which can go with 25 D regions. So the number of D to J combinations, let me draw it in black to emphasize, got to get the colors. You can get 25 times 6 D to J combinations. But each of these D to J combinations can go with any one of 50 V regions. So you can get a total of 50 times 25 times 6 recombinants from V, D, and J. So 50 times 25 times 6 is equal to 7,500. So from one gene, you can get 7,500 different heavy chains just by combinatorics. All right, but that's not all, because remember, there are light chain genes. And so there are 2 kinds of light chain genes. There is a kappa light chain gene and a lambda light chain gene. So the germline kappa light chain gene, kappa light chain, looks like this. It has a number of V regions and a number of J regions upstream of a constant region. So we're going to make that blue. All right. And so we have a number of V regions in the kappa light chain. Dot, dot, dot. V1, V2. And in the kappa light chain in humans anyway, there are about 40 of these. And then downstream of them, there are some J regions. And the J regions look like so. Dot, dot, dot. And there are 5 J regions. J1, J2, and J5. So how many recombinants can we get from the kappa light chain? For each one of the J regions, we can have 40 V regions. So there are a total of 40 times 5. But there's also a lambda light chain. Now it's interesting, the lambda light chain looks a little bit different. And actually, if you go to the chapter on gene duplication, at the beginning of the very origins of the VDJ recombination, you can imagine that the duplication could have occurred in a way to produce the lambda light chain. It's just a little bit of a different pattern of duplication. So the way the duplication happened was for the lambda light chain, dot, 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 there are a total of also about 40 V regions. Okay. And then there are 4 J segments, each of which is upstream of its own constant region. So the gene duplication that occurred looks like this. So here's one J region, upstream of its own constant region. I'm going to draw a different kind of constant region because this is now the lambda light chain. And then there's another J region. And dot, dot, dot. Let's just save some time here. Another J region. And there's a total of 4 of these, so J4, upstream of its own constant region. So as a result of this, we have a total, for the kappa light chain, we have a total of 40 times 5 possibilities. For the lambda light chain, we have a total of 40 times 4 possibilities. So the total number of different ways you can have light chains is 40 times 5 plus the number of ways you can have lambda, that's the number of ways you can have kappa light chains, plus the number of ways you can have lambda light chains. So this is kappa, kappa, and the number of ways you can have lambda light chains, which would be 40 times 4. That's lambda, okay? And that equals 200 plus 160. That equals 360. So, every antibody has to have 2 identical heavy chains and 2 identical light chains. There are a total of 7,500 different combinations that can produce a heavy chain, and a total of 360 different ways you can produce a light chain. The germline heavy chain gene undergoes VDJ recombination. And the light chains undergo VJ recombination. That's why the D is in parentheses, because heavy chain does the D, and light chains only do V and J. So the total number of combinations of heavy chain and light chain is 7,500 times 360. 7,500 times 360. And that equals 2,700,000. A truly staggering number. So just by combinatorics, 3 measly little genes, 1 germline heavy chain gene and 2 different light chains, kappa and lambda, 3 genes can produce 2,700,000 different combinations. That gives you incredible diversity in coding for antibodies. And as a result of that, we can fight off infections from bugs that we haven't even seen before, because we have that many different ways of making antibodies. Now contrast this to another strategy, which would be to make a separate gene for every different antibody that you could make. You'd have to make 2,700,000 different genes. Well, it turns out our genome really doesn't have enough room for that, because the human genome contains only about 20,000 genes. So there's no way you could have an immune system with this kind of diversity if you didn't use the combinatorics that are involved in VDJ recombination. So you've got 2,700,000 different ways to make antibodies out of 3 genes. And so you're only using up 3 out of your 20,000 genes to make antibodies. Pretty amazing. Thank you.